Good day, bioinformaticians. Now, in this video, we're going to discuss what is bioinformatics and the usage of one of the tools, Rosetta, in bioinformatics. Bioinformatics is the use of computers to characterize the molecular components of living things. Bioinformatics is a highly interdisciplinary field of biology, relying on basics from computer science, biology, physics, chemistry, and mathematics, statistics. The subject facilitates biological information to be handled by the use of computers. Most of the large biological molecules or polymers are ordered chains of simple molecular molecules called monomers. Monomers can be thought of as beads or building blocks that, despite having different color and shapes, all have the same thickness and the same way of connecting to one another. Each monomer molecule is of the same general class, but each kind of monomer has its own well-defined set of characteristics. Many monomer units can be joined together to form a single, far larger macromolecule, which has exquisitely specific informational content and or chemical properties. According to the scheme, the monomers in a given macromolecule of DNA or protein can be treated computationally as letters of an alphabet put together in pre-programmed arrangements or carry messages or to do work in a cell. So now, how do we explain the role or use of Rosetta in bioinformatics? Rosetta focuses on protein expression data and sets out to identify the specific proteins which are up or down regulated in a particular disease, and characterize these proteins with respect to their primary structure, post-translational modifications, PTMs, and biological function. Evaluate them as drug targets and markers of disease, and develop novel drug candidates, and use techniques called fluorescent IPG page to separate and measure different protein types in a biological sample, such as a body fluid or purified cell extract. After separation, each protein is collected and then broken up into many different fragments using control techniques. The mass and sequence of these fragments is determined with great accuracy using a technique called mass spectrometry, and the ratio computed thereof, that's m by z plus. Rosetta, then, which is built on an object relational database system, which stores demographic and clinical data on sample donors, tracks these samples which are processed and analytical results. It also interprets protein sequence data and matches this data with that held in public, client, and proprietary protein and gene databases. Rosetta comprises a suite of linked HTML pages which allow data to be entered, modified, and searched, and allows the user easy access to other databases. A high level of intelligence is provided through a sophisticated suite of proprietary search, analytical, and computational algorithms. These algorithms facilitate searching through the gigabytes of data generated by the company's proteomics projects, matching sequence data, carrying out de novo peptide sequencing in correlating results with clinical data. So Rosetta is something like the initial blast for protein sequences, to be specific. Thank you very much.